Right, so I'm going to start and share. There we go. Am I sharing? I'm sharing, right? Okay, can you see my screen now? I think it's okay. All right, so today, yes, we have...
Sylvie, sorry, we see the presenter view.
Okay, sorry about that. So click, I shared the wrong screen then. Share, and it's going to be this guy, I guess. Yes, now? Is it okay?
Now it's perfect.
Thank you. Okay, so yes, I'm going to talk about the anatomy of microscopes. So we're going to sort of dissect a little bit our microscopes today. Click. Right, so this lecture is a little bit big, and I broke it into 3 bits. And in between, we're going to do some quizzes so that we can relax our brains. So the first part of the lecture is called architecture, transmitted light versus fluorescence. So within microscopy is a very vast field, and it's a lot more than what we do. So within microscopy, we have, for example, material imaging that we are sometimes not very aware of. And scanning probe microscopy is another type of microscopy that also works with light. And we have electron microscopy, which doesn't work with light and has a very high resolution. And we also have medical imaging. And clearly, this is a very important field. And actually, the highest amount of money is in the material imaging, because this is when all the chips of all the goes into all the computers in the world are being scanned automatically to check for defect. This is the way it's done. It's done also with microscopy and automated image analysis. And then we have line microscopy for biological research, and that's what we are doing. So that's what we're going to talk about. And so within this field, we have several areas, and one of them is transmitted light microscopy, which is sometimes called bright field, and we will talk about that. And we have fluorescence microscopy. Okay. Within transmitted light, we can have a sample that is labeled or not labeled, in which case it is often called label-free imaging. And it's the same thing for fluorescence microscopy. Our sample can be labeled the way we do it, or it could be not labeled, and in which case we can look at autofluorescence, for example, or we could look like one of our students this year, look at reflection. And if anybody wants to know a little bit more about reflection, you're welcome to discuss. I can't remember. Who does reflection this year? Someone does reflection microscopy, right? Or you're imaging the laser light. We spoke about it yesterday. Pepe is not here. Okay, so and then we have so in transmitted light microscopy. This can be done on the wide field systems. It can be done on confocal systems as well, but not on light sheet systems. And so confocals, I will explain to you, but it encompasses the single point confocals and also the spinning disks. And then fluorescence microscopy can be done on any of these microscopes. Let me turn off my phone in case. I don't want disturbance. Yes. It is sometimes thought that the confocal microscope is for fluorescence microscopy and the wide field microscope is for transmitted light microscopy, but this is not the case. We can do both of them. Transmitted light is on a wide field system, but it's not true. We can do both transmitted light and fluorescence on both confocals and wide field. So transmitted light versus fluorescence, we could call this different microscopy techniques. Whereas wide field, confocal, light sheet, this would be different types of microscopes. We also have another way to differentiate the microscope is if they are upright, inverted, or flat. So we will talk about this. And this is more the architecture of the microscope. So now there is something that is already a little bit disturbing, and I think that when we speak microscopy, one of the issues comes sometimes from the fact that we do not know the words that we are speaking are not very precise. Let's say, like for example, when we talk about our fluorophore, which fluorophore we use, we're not very precise there, so we need to improve. So here we can see something a bit confusing when we have wide field and bright field, and these are two different things. So I just want, since I start talking about it straight away, to make it very clear. Bright field means a dark object on a bright background. Bright field is one technique of transmitted light microscopy. Okay, bright field, a bright background. Wide field means defocused illumination, wide. And we will see now how it works. So this is one type of microscope. Like we have confocal, we have wide field. Okay, so I will clarify this, but since I start talking about this, I want to make it clear from the start. What is the fundamental difference between all these? It's the light path. So what we're going to talk about today is very much the light path. So let's understand what the light path is. Basically, the light path always starts with a light source. And then we have optical elements. And then finally, we have a sensor. And it always ends with the sensor. So I want to, in this course, I want to make sure that we speak the same language. So I'm going to give some definitions that we're going to use throughout the course so that we all know what we're talking about. And that doesn't mean that these are the way people outside my world call things, but I think somehow we have to come to some sort of consensus for this course. I call in this course sensors any light recording device. So what type of light recording device do we have? We can have the eyes, for example. This is a light recording device. We have detectors on point confocals. We also have cameras. Okay, these are the major, or actually all of them, the light recording devices that we use in our field of light microscopy. And which optical elements are there? Well, for example, we spoke yesterday about lenses that we use to manipulate light. So yes, we have lenses, including the objective. We have lots of filters. So what type of filters do we have? We have excitation filters sometimes. So if we have lasers, we don't have them. And clearly, the excitation filters are often close to the light source. And because this is what they deal with, right? And then we have emission filters, and if we have them, because we could also have spectral detectors, and if we have emission filters, then they are close to the sensor, because they are dealing with the emission light. So this is all logical. We also have dichroic mirrors, and these guys are, so first, I forgot that. What are the three roles of a dichroic mirror? Can anyone tell me?
It directs the light to the sample.
Yes, it sends the light to the sample.
Yeah, and then it transmits the light that comes, so bounces off from the sample.
Okay.
And blocks the rest of the wavelengths that we do not want to reach to the sensor.
Okay, right. So we're sending the light to the excitation light to the sample. We are preventing the reflected light from going to the sensor. We're sending the emission light to the sensor. These are the three roles that the dichroic mirror has. And this is very important. We always need a dichroic mirror. So the dichroic mirrors are somewhere between the excitation filters or the light source and the sensor. So they are sort of in between there. We have pinholes in a confocal. That's also an optical element. So these are only in confocals, but we also have something that we should consider as an optical element. We have the sample, we have the cover slip, the mounting medium, and all these things completely are part of the light path. So a very, very simple definition, let's say. The world of microscopy is divided only in two parts. The microscopes that have a camera, that do not have a camera, and these are single point confocals, and microscope with a camera. And this is all the other microscopes, including wide field, including light sheets. So in that sense, it is quite simple. But these two are quite different, and we will have a look at them in more details. But first, I want to define things. What is a single point confocal? Because this is what I wrote here, single point confocal. Well, this is what everybody else calls a confocal, or a point scanning confocal, or a laser scanning microscope. So there are quite a number of names for it. But basically, it is a confocal microscope that scans the image point by point. I call this single point confocal, and you will see later why. Wide field, so I spoke a little bit about this. Wide field means defocused illumination, right? So it is very different from a single point confocal where all the illumination light is focused on one point. In wide field, we have a defocused illumination and the whole field is illuminated. And this is what people call standard microscope. Sometimes people call it an epifluorescence microscope or fluorescence microscope. And that does make sense because the confocal is also a fluorescence microscope and it's also epi. So, or a normal microscope, or sometimes people say simple microscope. Well, I think that it's not simpler than the rest. Let's call it what it's called for real. And this is called a wide field microscope. And this is not just my world. It's everywhere. It's called a wide field microscope. So this is what we should call them. So with camera and without camera, this is a very simple distinction that we have. And then we will talk more about this later. First, I would like to talk about upright, inverted, and flat architecture of microscopes. So when we come and we look at a microscope, we can see two different types of architectures. This is an upright microscope and this is an inverted microscope. What's the difference? Here, first, we can recognize the stage. So it's quite easy to see where the stage is. This is the place where we put our sample. And then we look at where the objectives are compared to the stage. So if the objectives are above the stage, this is an upright microscope. If the objectives are below the stage, these are inverted microscopes. So we can see that we have several objectives on this objective turret. It's called the stuff that turns. So it's called an objective turret. And we have several of them. But when we acquire an image, we only use one of them at a time. Okay, we can change objective. We only use one of them. Same thing here. We have several objectives. We can change them. But when we image, we only use one. On the other side of the stage, compared to the objective, we always have the condenser. So it's a little bit difficult to see here, but this part here is the condenser and this part here is the condenser. The condenser is always on the other side of the stage compared to the objective. So easy to recognize. Now let's look at an upright light sheet system. So the light sheet system is a little bit different in we can use several objectives to image at the same time. So some light sheet systems use only one, but most of them use several. It's very common to have two objectives imaging at the same time, to image at the same time, and sometimes you can have 4 of them as well. So this is an upright light sheet system. Why is it upright? Can anyone tell me?
Because objectives are on the top of the sample.
Exactly. The objectives are above the stage. Okay, so this is the definition of an upright system. The objectives are above the stage. If the objectives are below the stage, it's an inverted system. Good. And this is a flat light sheet. And why is it flat? Because this is the place where we put the sample. And when we look at this, if you would open this and you look from the top, this is what you see. So this is the place where the sample is. So that's the stage. And we have 4 objectives that are used simultaneously to image. And you can see that the whole thing is actually horizontal. So it's a flat architecture. Okay, so this is a lot less common. Of course, except for light sheet systems, the most commons are upright and inverted, and the most common is actually inverted in our field. And then in light sheet systems, we can have more variation like this. So what are the advantages of upright microscopes? Well, so the objective, upright microscope, the objective is always above the stage. That's the stage. The objective, the condenser, is always on the other side. And this is a little bit historical. So the first microscopes that were designed were upright. So the objective was above the stage. So it stayed like this for quite a long time, but it's very useful to image the top of a thick sample that cannot be flipped. So imagine that you have a mouse, for example, and you want to look at its brain, and you cannot flip it. You cannot. I mean, you could put it on its back, but it's not very physiological. So depending on what you do, you may not want to put the mouse on its back. And so you could also imagine that you can have a model, a tissue model, where that you're growing in the dish, and you have to look at it from the top. You cannot flip it. So because it has like sort of a top and a bottom. So this type of samples, you must have an upright microscope to image them. So also sometimes when lots of space is required around the objective and you also use transmitted light, then the condenser is always bigger than just one objective. So sometimes it's an advantage to have an upright microscope for this sort of reasons. And so upright microscopes are used quite a lot for intravital imaging. So for example, the brain of live animals, skin flaps, depending on where they are. And sometimes you can use like optical windows. And again, it depends on where they are implanted if you're going to use an upright or an inverted microscope. But these are, as you see, specialized reasons to use upright microscopes. The disadvantages is that because the stage is at the objective is at the top and it goes quite close to the sample, it's difficult. You're restricted in the stage movements. For example, you will not be able to use multi-well plates, and the objective will bump into the dish quite easily. So now inverted microscope. So as I said, the objective is below the stage. The condenser is always on the other side. So the condenser is above the stage. And oopsie, I went the wrong way. So this is much easier to use for live samples because you can use a CO2 chamber on top, and the objective is not going to bump into the CO2 chamber because it's below the stage. We can also combine, use the stage movement since the objective is not influenced by what happens above the stage. We can use multi-well plates and different type of dishes, so we have a lot more freedom to move the object compared to a and anyway have the objective very close to what we're imaging compared to an upright microscope. For that reason, most modern microscopes are inverted, and this is the case for most microscope in our field. We use inverted microscope. If you use a fixed samples between slide and cover slip, it doesn't matter at all. You can use an upright or an inverted, there is no difference. But for any sample that is a little bit bigger or a multi-well plate, and you anyway want to come close to the sample with the objective, so you have high resolution, it's not possible to do this on an upright system. So upright versus inverted, we have, so basically you can have them in both, in any type of microscope. This is like a choice depending on the experiment that you're going to have. They can do both fluorescence and transmission, and they can be fully automated in X, Y, Z, and wavelengths. You remember that I said that this is the abbreviation for wavelengths. So when I put lambda somewhere, it's always the wavelength of light. And then you can use live and fixed samples on both of them, and you can have very advanced techniques for both of them, like FRAP and FLIM and all this. However, the upright system is best to image the top of thick samples that cannot be flipped, and the inverted system is best for everything else. So this is the reason why there are many inverted systems, is because of the flexibility. Another difference between upright and inverted is that we do not have any hardware autofocus on an upright system. So we will talk about hardware autofocus a little bit later. And it's a very useful tool that we cannot have them on upright. Good. So now we're going to talk a little bit about the light path for these two types of systems. So we have again our upright and our inverted system. And just to remind you that the objective is above the stage for the upright and below the stage for the inverted, and the condenser is always opposite. The emission light path is the part of the light path that goes from the sample to the sensor. And mostly this goes through the objective. So we're imaging through the objective. So the emission light path is sample, objective, sensor. So you see, we're following the light as it goes through the microscope. This is what the light path is. So for this reason, because we go sample, objective, sensor, the sensors are mostly on the same size as the objectives. So that is a quite easy way to recognize where a sensor is. You look where the objectives are, and you look at the same side, and you should not look on the other side. This is the case. This is true for almost all cases. I will talk about the exceptions later. So on this upright system, we start from the sample, and we say, okay, we go through the objective, right? Because it's always sample and objective. Okay, fine. The emitted light will go through the objective, and then it keeps going, and it will reach this sensor, which is a camera, or it would reach this sensor, which is the eyepiece, in case we want to look with our eyes, which are also sensors. Okay, so this is the emission light path, very simple on this upright microscope. And we look at it on the inverted microscope. It's also very simple. So we start from the stage, we go to the objective, and then we keep going. Here, there is a mirror that directs the light towards this, which is a camera, or towards the eyepiece, which is here. So this is the emission light path, quite simple. But there are some exceptions, and there are only two exceptions. One of them is multiphoton microscopy, and the other one is when we image transmitted light on a single point confocal. So we'll talk about this so that we understand how we do that. Okay. So what about the excitation light path then? It's quite different. So the emission light path is always the same, almost always the same, except these two exceptions. But the excitation light path can be quite different. So we're going to look, there is a difference between transmission and fluorescence, and a difference between confocal and wide field. But first, we're going to do a little quiz. So you guys go to Glick, if I manage. So here, if you guys go to Canvas, and we will, I will bring this in so for people, and you look at this quiz called Microscopy Architecture, and then you can do that. Oh, that's not what I wanted to bring in. This one. So. Right. So you guys you can do it on Canvas, and then sorry, it's that one, and then I will just show it here. People are interested online. Okay, so this is the first question. This microscope, is it upright, inverted, or flat? And also, where is the condenser? Is it above the stage or below the stage? Okay. Was it okay? So where is the condenser in here? Anybody can tell me?
Above the stage.
Okay, it's this guy, right? This is the condenser. We can bring it up and down. Okay, and was that an inverted or an upright then?
Inverted.
It's an inverted microscope. Great, well done. Now we keep it going. Okay, so we have seen now the difference between inverted, flat, upright, and flat architecture. And we have seen also the emission light path, which is quite simple. And now we're going to continue, and I will tell you what I mean by microscope and satellite equipment. So this is a microscope that is at the Life Cell Imaging Core Facility. And we have here the microscope. So we often say, oh, this is my microscope, but actually the microscope is only this. It's the microscope body. Everything else is satellite equipment, and that's a lot. So there's a lot more satellite equipment than the microscope. And these satellite equipments, they are attached through different types of connections. So they can be attached through cables. So if we look, zoom in a little bit to see, we can have cables like this that are attached to the satellite equipment. Or we could have these fibers, which are like metal things that you may have seen sometimes on your microscope. And these are optical fibers. Or we could have hard connections like this. Oh my God, she wants us to learn everything. No. So the good thing is that you only need to care about the equipment that is attached to by hard connections. Once you recognize if you learn how to recognize the equipment that is attached through hard connection to the microscope body, then you can recognize any microscope. And that's what we're going to do today. So one very important tip as well is, as we said before, the sensors are mostly on the same side as the objective. So the first thing that you do when you come in front of a microscope that you have never seen, if you want to impress your colleagues and say, oh, this is a spinning disk confocal, you can look at if it's an upright or inverted. And if it's an inverted system, the objectives are below the stage. And so the sensor must be somewhere below the stage. Okay, so this is an inverted system and this is a camera. So this is the sensor. So, and then if it's an upright system, you're going to look for the sensor at the top. So that's a pretty nice tip. So we're going to do this afternoon and learn all the tips and the tricks in the Anatomy of a Microscope workshop. But first, we're going to look at the satellite equipment that is attached by hard connection to the microscope body. And there are not so many options, so it's quite nice. And I remind you that the world of microscopy is only divided in two categories. The microscopes without camera, so of course some of them cannot image at all, but we don't care about those now. And then we have those that do not have a camera, but anyway can acquire images, and these are all single point confocals. And so I remind you, this is like what people call a confocal, and we'll see now why I add the single point. And then we have microscopes with a camera. All the other microscopes, apart from single point confocals, have a camera. I think this year we have about 50-50, a little bit more than 50% of the students who use single point confocals, and everybody else uses microscope with a camera. So now if the microscope has no scanner, it's a wide field system, and we will learn how to recognize that. And if it has a scanner, it's a multi-point confocal. And so what is a multi-point confocal? This is what everybody calls a spinning disk. And I think that it creates a confusion because people do not realize that a spinning disk is a confocal. So I prefer to call this a single point confocal and that one a multi-point confocal. And I think it clearly says what it does. They are both confocals, but this one images one point at a time and this one many points at a time. So we will talk about all this later. And there are a few more things, but mostly we need how to recognize a camera, how to recognize a scanner, and then we will be able to identify wide field, single point confocals, and multiple confocals. So now we're going to talk about the difference between transmitted light and fluorescence. So what is transmitted light? Transmitted light is often called a bright field. And I've told you before that bright field is called that way because we have a dark object on a bright background. And this is a beautiful example from Waldo Nell. I took this from his website. I asked him. And these, you can see that the object is dark and even the little line here are dark compared to a white background. So this is our light background. This is called bright field technique, and it is not labeled at all. So the light goes through the sample. This is why it's called transmitted light. Transmitted light, the light goes through the sample. That means that the sample must be transparent or thin. And if you wonder why we are cutting our tissue sections as thin as possible, let's try to do 4 microns, let's try to do 5 microns, even though sometimes it doesn't make sense to cut them so thin. It is because we have inherited this from the hospital because they use a lot of transmitted light microscopy. And so they have to make the sample transparent so that the light goes through. And so we continue doing this even though we use fluorescence. So yeah, we do inherit a lot of things from the hospital. So transmitted light imaging is available on wide field system and confocal systems. So the only type of microscope that doesn't have any is the light sheet. So wide field, what does that mean? I'll remind you that this means defocused illumination, and this is what people call a normal microscope. Well, it's called actually a wide field microscope because the illumination is not focused, as opposed to a confocal where it is focused. So we have wide field and bright field, and please don't confuse them anymore. So transmitted light, the light has to go through the sample, and it is a low contrast type of imaging. So it's a little bit like looking at the stars in the sky during the day. It's difficult to see them because we have a light background, and therefore there is a low contrast. The contrast can be enhanced in transmitted light imaging. I'm going to remove this guy's stick. The contrast can be enhanced using some optical elements. And so the very common ones are a DIC, differential interference contrast, or phase contrast. And you can see here the difference that the background becomes darker, and therefore we have a higher contrast. So we have enhanced the contrast. So this has colors, but this sample is not labeled at all. This is due to the phase contrast that we see slightly different colors in here. And so we are enhancing the contrast with optical elements. Yes. So we get a higher resolution, but in any case, all these techniques, transmitted light techniques, they have a low contrast. So even here, we can say we do not have a very high contrast. And we have no molecular specificity because we do not label anything. So, so no molecular specificity for this. The contrast can be enhanced using colored dyes. So this is an example of a sample that has been labeled this time. So this is not labeled, and this is labeled. And it has been labeled with 3 different colors. So there's blue and red and brown. But there are only these 3 colors available, so we cannot have more. So these dyes that we're using, they have a color. They're not fluorescent. They're called chromogenic dye. Well, some of them are also autofluorescent, but they actually have a color. So chromogenic dyes, they require a color camera to be imaged. So otherwise, in general, we image with black and white sensors, and we're not going to be able to see the color, the difference in color between these dyes. So we need a color camera. So the applications for transmitted light imaging are that you can put the fluorescence in the context of the unlabeled tissue or cells. If you think about it, when we image fluorescence, we only see what we label. And that can be a great disadvantage. So it is really good to take a transmitted light image as well as fluorescence and overlay this so that you can see the context of the fluorescence. So that is a very, very useful application of transmitted light imaging. Also, it's used a lot in the clinic because the microscopes are cheaper, basically. So it's used a lot at the hospital. And this is label-free microscopy. This one is not because we have labeled, but this is actually label-free microscopy. And then there is an extra little technique called dark field that is not used a lot in where actually we have a darker background. And it's not used a lot in microscopy in our field, let's say, but it's used a lot in material imaging, for example. So this is an example of what we can do as well with transmitted light microscopy. So it's definitely, you should not look down on transmitted light microscopy because with the advance of AI, it is fantastic what we can do. You can see here how we can track these cells and like pretend that we have a label on the nucleus. This is a fake label that is found by AI. And it's really extremely powerful. It's very low invasive. So the light is very gentle when we image, and we do not label these samples. So we really have a very low impact on the sample when we handle them and when we image them. And you can see in here, even this very complex pattern in here has been identified by AI. So they can AI. So with a good trained algorithm, you can figure out where the compartments are. So all these are really, there's a lot of advances nowadays with transmitted light microscopy and AI. So what is the light path for transmitted light microscopy? So I remind you, the light path goes from the light source all the way to the sensor, which can be the eyes, the camera, or a detector. And in between, we have all these optical elements. So this is an inverted system. And the emission light path is mostly sample, objective, sensor. So here is what we have seen before. So we go from the sample, which is on the stage, to the objective, and then we continue. Here, there is a mirror that sends the light to the sensor, which should be here, and it would be a camera, but it's actually, I think, on the other side. And in here, we could also send the light to the eyes, which are also a sensor. So this is the emission light path. But where does the light come from? So when we look at the excitation light path, we go from the light source to the sample. So there are a few rules. In transmitted light microscopy, the light always goes through the sample. So if the sample is on the stage and the sensor is below, then the light source must be at the top. And you can see here the light. This is the light source. So the sample absorbs some light, and the rest, whatever goes through the sample, is transmitted through the sample, and it's imaged on the other side. So we always have, the light source is always opposite compared to the sensor, because it has to, the light has to go through. So light source and sensors are always on opposite sides of the sample. Okay, so here we have the light source. It goes through the condenser, which you know now. And we go through the sample to the objective, and it goes down, and we send it either to the camera or to the eyes. And the light always goes through the condenser, okay? So in most cases, the light source is a halogen lamp or a white light LED. Nowadays, we also have that. So someone was asking yesterday, I think, about the halogen lamp. This is what the halogen lamp on your microscope is used for. It is used to image or look at transmitted light. So the light source is opposite the objective. So we have the objective here. The light source is at the opposite the objective on the other side. So there's only one exception, and that is when we image transmitted light with a single point confocal. So we will see this because quite a lot of you have single point confocals, and we need to learn how that works. That's the only exception. And in that case, when we image transmitted light with a single point confocal, the light source is the laser. So we're using the lasers to image. So this is also used for fluorescence imaging. And the light source is on the same side as the objectives. And then the lasers are sent through the objective, because in transmitted light, the light always goes through. So the light goes through the sample, and it goes to the condenser, and then it goes up here. And that means that we have to add an extra sensor here. Okay, the light source and the sensor have to be on opposite side of the objective because the light goes through. So in that case, the light source is on the same side as the objective and the sensor is opposite. But in transmission, in any way, the light always goes through the sample. That's why it's called transmission. Okay, so here we have the two light paths. If we use a point confocal when we want to image, we have the light source here, it's the lasers, and it goes through the objective, through the sample, the condenser, and to the sensor. And if you use any other type of microscope, the light source is going to be here for an inverted microscope, and then it goes through the condenser, through the sample, through the objective, and then to the eye or the sensor. So that is the light path for transmitted light. So here we have a little zoom poll. So now this is quite special. Zoom poll. How do we do that? Let me see. I think I have that somewhere here. Okay, launch the zoom poll. So can you image, can your microscope image transmitted light? Or do you not know if it can?
I think panelists cannot vote based on what I can see.
Really?
Yeah.
Yeah, it says that host and panelists cannot vote.
Oh, that's interesting. Okay. Sorry about that. Okay, it seems that many people online have, okay, it's quite bizarre. I will have to have a look at this. But you can tell me, can your microscope image transmitted light? Does anyone have a microscope that cannot image transmitted light? I know at least one. Yes, Anne-Marie has a light sheet system. It cannot image transmitted light. And does anyone not know if their microscope can do that or not? Oh, okay, good. But it's but similar online. Basically, we have most people say that yes, it's possible, and also some people do not know. So that's also completely valid. So if I go back here and I say, how can I do that? I do not know. And Paul, yes, lunch. So what about the second question? Can you tell me if you have ever imaged transmitted light? Has anyone never imaged transmitted light on their microscope?
I think I have never. At
Okay.
least on the one I'm using now, it's the spinning disc. So
Yeah,
that's why I was a bit unsure, but I'm guessing I can.
yeah. No, absolutely, you can totally, yeah. But and then anybody else has never imaged transmitted light? So in this course, you will have to. So you'll have to give us some images or transmitted light. So you'll have to go and ask the person in charge. So just think about it already now. That it's one of the requirements is that you do the color illumination for transmitted light. So you will have to do that. Okay, and yes, I think quite a lot of people have image with transmitted light. I think it's important to think about it. Transmitted light is it's very good to put the fluorescence in context, so that will help you. We're also going to do a little quiz. So let's do the light path quiz, which is right afterwards. So you guys can do it in Canvas, and then I will show it anyway. Okay, so using your favorite microscope, you can image transmitted light. You can see that the light comes from the top of the microscope, so it reaches the sample from above. Is the sensor above or below the stage?
Below.
Okay, below. Anybody? Why? Anyone can say why this is below?
Because in transmitted light, the light goes through the sample, so the sensor has to be below the stage.
Exactly, because it goes through the sample, so we must have the light source on one side and the sensor on the other side. Good, we're on track. So now I will continue. It's a little bit of a gymnastic to start thinking in those terms, but it's actually really useful. So I think you will get used to it quite quickly. So now we have spoken about transmitted light microscopy. Now we're going to talk about fluorescence microscopy. And I need to also keep track of time, I think. So I will have a quick look at this file here. And right. Okay, I'm good. Oh, this is not so good. Oops. Okay, so now we're going to talk about fluorescence microscopy. And one of the advantages of fluorescence microscopy is the contrast. So this time we have bright objects on a dark background. And so in fluorescence microscopy, we spoke about it yesterday, we always have two lights involved. So there are two light sources. So we have the excitation light and the emission light, and I mean we have two light sources. So the excitation light is very strong, and it comes from either a lamp, like a mercury lamp, LEDs or lasers, and it goes to the sample. And then the emitted light is very weak, and it comes from the excited sample. So the sample acts like a light source, And then it goes from the sample to the light source, to the sensor. So the fluorescence microscopy is available on all types of systems that we use, or it can be available, of course, clearly if it is equipped for that. And it is very much like, it's a very high contrast technique. So it's a little bit like seeing the stars in the night sky. We are going to see lots of small details that we are unable to see during the day. So if you think about it, during the day, the stars are still there. But the only reason why we cannot see them is because of the contrast, and it's not because of the resolution. And so we have to start thinking contrast. Resolution is very important. Contrast is also super important. If you have no contrast, you can have as much resolution as you want. You will not be able to see anything. And that is something that we will talk a lot about in the next week and even tomorrow. So the contrast can be further enhanced in fluorescence microscopy using different colors. So even if we have two objects that are very close to each other, if they have different colors, then we can differentiate them. And in fluorescence microscopy, we use mostly black and white sensors, and that is because they are more sensitive. We can use color sensors, but then we lose a lot in the sensitivity. And therefore, we need to have a way to separate all these colors. And as we have seen yesterday, we use filters to separate the different colors, filters and dichroic mirrors. And we do a good job so that we are sure that we don't have bleed through. So the applications of fluorescence microscopy, anything that requires a high molecular specificity. So we can, for example, differentiate between a molecule that is not phosphorylated and the same molecule phosphorylated. So this is a very high molecular specificity. And the resolution is high and the contrast is high. So I remind you that in transmitted light, we either have a high molecular specificity, because we could also use these antibodies against phosphorylated proteins or not. So we have a high molecular specificity or a high resolution and contrast. So we don't have both of them. We have to choose, basically. So here we have the light path for fluorescence. So we're going to look at it. The light path is always the same thing. It starts from the light source. It ends at the sensor. And then in between, we have the optical elements. So there are a few things that are always true from any fluorescence microscope. The light source delivers the excitation light, and it's either a mercury lamp, an LED, or a laser. On this side, the objectives are below the stage. This is an inverted microscope, and the light source is going to come from here. The excitation light is delivered through the objective. Okay. And the light source, and therefore the light source and the objectives are on the same side, right? So it's quite different from transmitted light. It's actually, I shouldn't have taken this image because we can still see the transmitted light. I used the same image as before. So we have the light source here on the same side as the objective. And then when the sample gets excited by this light, it fluoresces in all directions. And some of this light is going back through the objective. So in most cases, only the emitted light that goes back through the objective is detected, or we can look at it with the eyepiece in most cases, but there are exceptions, and I will tell you what. So it's both the excitation, in fluorescence microscopy, both the excitation and the emission go through the objective. And that means that the other side of the microscope, where the condenser is not used at all. So when you go to your microscope and you image or you look in the eyepiece at fluorescence, you can try if you want. If it's an inverted microscope, you can try and flip this arm. In general, you can like tilt it. You can tilt this and the image will not change at all. The top of the microscope on an inverted system, when you look at fluorescence, is not used at all because both the excitation light and the emitted light go through the objective. And so there is only one exception. When we image with a multiphoton laser on the multiphoton microscope, we have the possibility to also catch the light that is escaping, let's say, and is not going back to the objective, we can put a sensor here, an extra sensor, and then we can grab this light. This is the only exception. Otherwise, every time we image fluorescence on any microscope, we have the upright or inverted, we have the excitation and the emission going through the objective. So this is, yeah, exactly. In most cases, the excitation and the emission light go through the objective. So here we have the light path. We have, so the lasers, that's also something. The lasers are never used when looking in the eyepiece. So we always use either mercury lamp or LEDs. So this light, which is not lasers, will come in here. It will go through the objective. The sample will fluoresce, and the fluorescence that goes back into the objective will then be sent to the eyes, and that cannot be a laser. So can you tell me on your confocal microscopes, what type of light sources you have? How many light sources do you have on your confocal, point confocal microscope? Anyone?
I mean, we have a. I think we have a LED for the bright field. We have. I can't remember which kind of lamp because I don't use it often. But it starts with H.
A halogen lamp, HD.
A halogen lamp. Yes, thank you.
Sorry, not or halogen. I think you have an LED maybe for fluorescence,
Yeah.
and you have a halogen for transmitted light.
Maybe, but the lamp we use for the bright field is like inside the microscope itself. It's not.
Yeah.
While the halogen lamp is a box next to it. So.
Yeah, so I think you have a halogen lamp for the halogen lamp is always for transmitted light.
Okay.
So it's either a halogen lamp, or if it starts with H, it could be also a mercury lamp because it's Hg, mercury for some reason. But I think it's likely to be a halogen lamp, and that you use for transmitted light. And then you have lasers, of course, right?
Yeah.
So on the point, on the single point confocal, we have usually 3 types of light. We use the lasers when we want to image anything, transmitted light or fluorescence. And then when we want to look in the eyepiece, we anyway need a halogen lamp because we cannot use the lasers. And we can also use, and we also need like a LED, for example, to look in the eyepiece at fluorescence. So on the single point confocal, we're going to use a different light source if we look in the eyepiece or if we image. That's not the case for other systems. That is because the laser light on a single point confocal is very focused into one point. It would be very dangerous for you to get it into your eyes because of this very high power. And so this is not the case in wide field microscopy, for example. So we are going to become expert at all this today. Right, so now if we summarize a little bit, we have the transmission versus fluorescence, and we have spoken also about inverted versus upright, and there are 2 exceptions. So let's have a look at the light path. This is a summary of all the light paths possible. So we have here transmitted light, and here we have fluorescence. And we can have the microscope can be upright or inverted, right? So here we'd say we're doing transmitted light, but we have an inverted system. And here we are doing fluorescence, so we have an inverted system. And you can see that it's inverted because the objective is below the sample. And our sample is a little red fish. And the objective is pointing upwards, so this is inverted. That could be also upright. So in that case, the objective is above. So we have our sample and above, and we can also do fluorescence microscopy or transmitted light microscopy. And the condenser is always on the opposite side. The condenser is only used when we do transmission microscopy. The condenser is not used when we do fluorescence. So sometimes it's there, but we do not use it at all. And the sensor is mostly on the other side of the objective because, as we have said, the emitted light will go through the objective. So we must have a sensor on the other side to capture this light that has gone through the objective. So here we go. On an upright system, the objective is above, so the sensor is at the top. And on an inverted system, we have the sensor is at the bottom. So again, the same thing. The fluorescence will go through the objectives and to the sensor. In transmitted light microscopy, the light source and the sensors must be opposite each other because the light has to go through the sample, right? So here we have the light source. So in transmitted light microscopy, we have the light source in an inverted system. The light source is at the top. The light goes through the condenser, through the sample, through the objective to the sensor. In an upright system, it's the opposite. Light source is at the bottom, and then condenser, sample, objective, and sensor. And in fluorescence, it's quite different because the light does not go through the sample. We actually have the excitation and the emission light go through the objective, both of them. So the sensor and the light sources are on the same side for most cases, okay? And there's only one exception to this, or a few exceptions to this, only 2 exceptions to the whole list here. So the imaging transmission, when we want to image transmitted light microscopy on a single point confocal, we use lasers. And so when we see here on an inverted system, we are using lasers, so they have a color, that's why I put it in green. And the light, the laser light is going to go through the objective, through the sample, because this is transmitted light microscopy, it always goes through to the condenser, and then we add an extra sensor opposite. And if it's upright, it's the opposite. So that is one exception, and this is when we image transmission on a single point confocal. So that is a little bit special, let's say. And then the other one is when we image fluorescence on a multiphoton with a multiphoton laser, then we can capture the light that does not go inside the objective. So we could add a second sensor for the light that doesn't go back, the emitted light that doesn't go back through the objective and that escapes. We can capture it as well. So it's the same for an upright or an inverted. So these are all the possible cases that we have. And really, your systems are very much either this for transmitted light microscopy, this is a point confocal, and this is any other type of microscope. And here for both single point confocal and any type of microscope, it's the same thing. Both the excitation and the emission go through the objective. So at the end, we end up with very few configurations that apply to all the microscopes that you have in this course. And so we're going to go through more details with this, but this is a good summary of what we have in this course. Okay, let's go for a little quiz again. So this is the quiz fluorescence versus transmission. You can have a look at here. And if I go back to my module.
Hi, can I ask something?
Yes.
So in the single point confocal,
yeah,
the transmission light source is the laser. But if the microscope, let's say, had another light source on the top of the microscope,
yes,
then the detectors that are below the objectives could be used for transmission, right?
No.
No.
No, and that is because of the way the light has to be scanned. The emitted light has to be scanned. We cannot use a halogen lamp to image on the single point confocal. So what we do instead is that we add a little detector opposite the lasers. So this is why you have the detector in transmitted light microscopy on a single point confocal. You have an extra detector. Is your system inverted or upright?
Yeah, inverted.
Inverted. So you have so you have a detector which is often called a TPMT.
Yeah,
So
I have to check the pictures that I took. But
yeah, exactly.
probably.
So we will see all this and we will see the light path. But basically you will have in an inverted system because the light source comes from underneath. So the objectives are below, right? So the light source comes from inside the objective and it goes through the sample and then it keeps going. So it goes through the condenser, which is above the stage. And then we add an extra TPMT and a T there is there for transmission. So transmission PMT, so it's a transmission sensor. And that is because of the very peculiar way a single point confocal works, that it images one point at a time. So we have to use anyway some condensed light, even for transmitted light microscopy. So we'll see all this afterwards. I'm sure you will discover lots of things about your microscope. So here we have a little quiz. So you can do that. Well, we're not going to do that in quiz. Let me see a little bit that I. Right. So now we have a few quizzes. I think we're going to go to the workshop room to do that, because I want to be able to put you in groups, and it's not possible on Zoom. On this Zoom. So we go to the workshop Zoom, and then we will be back at 10:30 because we have a little break as well. So we do a bit of quiz, then we have a little break. So we'll be back in 10:30 in here for the second lecture.